14th Biannual Conference. We are in the sub-theme Transformative Agri-Food System. We'll be handling plant, crop sciences, as well as soil science presentations. I want to welcome all the participants. For the presenters, you have 10 minutes of presentation. If you can do less, the better, and 5 minutes of question time. So the first presenter is Mr. Chungwa. And he's going to present on development of somatic hybridization protocols for taro. I welcome Mr. Chungwa.
Sorry, if you may allow me to interject. For some reason, Mr. Chungwa is not able to give the talk, although this is his work. And just because I'm part of the team, I'll just take the opportunity to give the talk. Just to apologize that Mr. Chungwa is not able to give the talk.
Okay.
So I'll take over and give the talk on behalf of Mr. Chungwa.
Welcome, Mr. Boal.
So let me share my slide. I guess everybody can see my slides, just to be sure that we are on the same page.
Yeah, we can see. Can see your...
Okay, thank you. How do I go to maximize my screen? Okay.
Just put it on the presentation board.
All right, so let me just do the presentation mode and hide this. So I'm told that the talk has to be 12 minutes, so I'll try my best to give the talk within 12 minutes. So just to give you a quick background about this work is for an MSc student, Mr. Chungwa, and Mr. Chungwa is being supervised by Professor Gesimba and I, and he's a student at CHS, Egerton University, that is the Crop, Horticulture and Soil Science Department. So this is part of the work for his MSc, and he was supposed to give this talk, and for some reason we had some miscommunication. And I again repeat, I just apologize for this miscommunication, and I think the fault is on us as supervisors. And when our students are down, I think the honors is on us to pick them up. So I'll start by introducing this talk by saying that this talk is about a technique that has been there for quite a long time. However, this technique has not really been taken advantage of, especially when it comes to indigenous crop species, especially the root crops, those which are actually regarded as being neglected. So we are trying to put together a protocol to do what we call somatic hybridization. Basically, in simple English, it means taking two plant cells and putting them together. And the idea of working with taro using this technique is because of the fact that taro really has a problem when it comes to producing flowers. So this is just one step into many opportunities that we want to open up, especially when it comes to research on taro, and here particularly in Kenya. So most of us are familiar with taro, and you may want to ask yourself if indeed you know taro, and majority of us, especially here in Kenya, we call taro nduma, and sometimes we call them arrowroot, which is actually a misnomer. Arrowroot belongs to the family. Is actually a root crop that is known as Maranta arundinaceae, and these actually forms a rhizome, while taro or nduma is actually a root crop also that forms a culm. And this is just basically how taro looks like. This is just a field of taro crop, harvested taro crop, and of course there are many, many products that come out of taro. And again, people consume leaves, especially when you look at literature from Hawaii and Asia Pacific. However, in East Africa, yes, the Indian community, they use these leaves in terms of, like a vegetable. So basically, nduma or taro is a root crop that is well known in Kenya, and of course it has got its own problems, especially when it comes to production. And of course, disease and many, many other constraints, availability of planting materials, etc. Now, because of the problem that is associated with taro in terms of flowering, we thought of an idea of how we can solve the problem. There are various ways, or if you like, approaches that have been, used, or if you read literature, there are those who use plant growth regulators to sort of force the taro to produce flowers. And there are quite a number of groups which have reported, especially if the one that comes to my mind is quickly is a Japanese scientist called Naoki Katasura, who tried to use GA or gibberellic acid to induce or to force the taro to produce flowers. Yes, my slide says taro rarely produces flowers, but there are certain species which have been, known to produce flowers. And just for the sake of those who have not seen taro flowers, it looks like, the flower, the images on your left side, on my left side of the screen. These are an open flower, and the flower here we have the female part and the male part, and of course, when there's fertilization, you have now the seed pod forming. So we looked at literature and we found that inducing flowers might not be a good idea, especially when it comes to opening up more opportunities for taro improvement. So we decided to move towards protoplast. And because of the fact that protoplast is a great way in terms of opening up new opportunities, especially for taro, which is regarded as a neglected root crop in Kenya. So by working out or developing protoplast fusion protocols, it will help us also in future to produce more propagule, because vegetative propagation, if you require, for example, 1 million seedlings, then the easiest way to do it is through biotechnological approaches. And protoplast is one way in which you can produce also propagules. Now, you can also use protoplast to open up opportunities such as genome engineering. Now, I know many people would want probably go the easiest way, use tissues, whether it's leaf, color, etc., to do genetic transformation. But the dangers lie in the fact that when you're using tissues, then you might regenerate what we call the chimeras, meaning the roots might be transgenic or might be genetically modified while the leaves are not. So when you start with single cells, and especially protoplast, so the moment you engineer changes in the protoplast, then it means if you regenerate the plant, or the whole plant will be genetically transformed rather than part of it. So it opens up new opportunities for taro. Apart from just genetic modification, we can also do the cell hybridization, bring in the two cells, which is the core, or if you like, which was at the heart of this project for the MSc students. So protoplast opens up new opportunities, and that's why we share the idea of using plant growth regulators to induce flowers. So this work was conducted in several phases, if you like, which I'm calling method. There are 3 of them, meaning stepwise, because we decided that we're going to use leaf as source of protoplast or
Recording in progress.
Plant growth regulators, especially when you are talking about the 2,4-D and TDZ, which were the excellent, really gave us embryonic color. Now, the second step of the project for these MSc students was again to now do the actual isolation of protoplast. Again, we looked at 2 different species, that is the Egerton, and of course we used also 2 explants, that is the leaves and the embryonic color that we generated in method 1. We tested enzyme incubation period because we did use a combination of both the pectinase and cellulase, and we used different time periods to see which time period is excellent in terms of production of the protoplast. Of course, at the end of each of these incubation periods, we indeed looked at the quality of the protoplast that we are getting. So we also ended up looking at the quantity, that is the amount of protoplast we are getting, and of course the quality that we were getting at each of the time period.
Hello, Mr. Guo.
Yes?
Your time is almost up. 1 more minute to wind up.
All right. So we also did now the actual fusion. So we took about 3 fusogen. We tested the 3 of them. And just to quickly look at the results, we of course found that 2,4-D and TDZ was excellent. And of course, at either the level of 2 and 4 milligrams for 2,4-D and in combination TDZ was excellent, was giving us good quality. And when it comes to production of the protoplast, again, we realized that there was a trend. The longer we incubated, the lesser we got the yield. And of course, the leaf tissue was giving us the best in terms of the protoplast yield and quality, unlike the Carli. Now, we also found that we also found out that for the fusogen, actually PEG and calcium chloride were giving us at least good numbers in terms of the fused, protoplast. But overall, the PEG 6000 was the excellent fusogen when you do it at 20 minutes. So we observed quite a number of, phenotypes, if you like, in quotes. We had some cells, 2 cells fusing, that is from the leaf and also from the protoplast. We also had leaf to leaf and all that. So we've got quite a number of fusion, either binary or multiple fusions that we observed. And of course, in the end, we realized that when you incubate enzymes, especially when you're producing the protoplast, the longer you incubate them, the more or the less you get viable protoplast. So time period is quite important when it comes to yield. And of course, explant type and the genotype, that is Egerton, really influence the yield of protoplast that we're getting and even the quality. And of course, when it comes to fusion, going more than 20 minutes, especially for the best fusogen, PEG and calcium chloride, then you don't get quite good numbers in terms of fusion. And of course, in the end, we realized that after we did the fusion, now getting the actual microcalli was really a problem. And of course, MSc project is time bound. So we had to allow the student to start writing so that, he can graduate. But we feel that we need to proceed again, follow up this work, and of course, optimize the media for culturing the fused cells so that we can get plants at the end of it, which is what is more important for us. Of course, we got funding from quite a number of organizations, NRF Kenya, which received funding from NRF for this bigger project for Taro. And of course, the student also was under SESAM, and most of the work that I've reported here was done by the student in a lab that is funded by the Confucius Institute at Egerton, that is the crop molecular biology lab. Thank you.
Thank you very much, Ms. Dr. Boao, for that presentation on behalf of Mr. Chungwa. We have some questions on the chat, and you can respond to those. But I give an opportunity just for one question, one burning question to Dr. Boao. Any questions? All right.
Okay, just to answer Steve Gizingu's comment or question, we didn't manage to generate plans, but that was the ultimate goal, and because of the time, period for the MSc student, we feel that this is something that the next student can pick it up from. So we didn't manage to generate plans, but we got microcalli, and of course this means that we have to play around now with the media formulation so that we get allow the protoplast to fuse, get the cells together, get the microcalli, and then again regenerate plant. So this work is still ongoing.
Thank you very much for the time that you've given in the presentation. Now, our next presenter is Munyoki. Is Munyoki in?
Yes.
All right, you're welcome. You have 10 minutes. Please keep time, give the very essentials of your study so that we don't harass you in terms of movement, in terms of time. Just the key points or key observations that you made in your study. Welcome.
Okay. I hope you are hearing me.
We can hear.
I'm trying to attach my document.
Put it on presentation mode. Slide mode. Okay.
Okay, I'm James Ngelezi, a PhD student in the Department of Crop Horticulture and Soils. And my supervisors are Professor Munaru Said and Professor Joshua Gwen. I was working on integrated management of root node nematode using fresh manure and the cropping regime for improved growth and yield of tomato. Next. So on the introduction part, you find that tomato is a relatively short duration vegetable, making it an economically attractive crop to many farmers. You find that tomato, despite being an important crop, the production still falls below the demand of the produce, and tomato production can be increased either by increasing output per unit area of land or putting more land under production. The next slide. So you find that arable land, especially in Kenya, is diminishing by the day. So you find that the former remains the viable, whereby production per unit area is the one we can use. We can increase production per unit area because the land is diminishing, and you find that production under this unit area can be maximized through proper cultural practice and also pest management. You find that tomato production among many farmers is affected by factors which include biotic constraints, and some of them are root node nematode. Next slide. So currently you find that control of root knot nematode is controlled the previous slide. You move back, previous one.
Previous slide.
You find that currently the control of root knot nematode are being controlled by use of chemical, nematicide, and crop rotation. And you find that chemical nematicides are currently being replaced with respect to being hazardous to the environment and also human health. On the other hand, you find that crop rotation is currently not feasible within smallholder farmers due to their limited land resources. Next slide. So you find that little attention has been paid on how underground seed plant, intercropping, and organic manure could draw root knot nematode under our local conditions. So you find that in this study, we evaluated effects of integrating fresh manure and slender leaf intercrop as an alternative method of managing root knot nematode, as well as establish the effect of the strategy on how they affect the growth and yield of the tomato. Next. So this study was carried out at Horticultural Field 3, which is a research and teaching field at Egerton University. And the experiment was 2 by 3 bacterium, which was arranged in an RCDD with replication. And the two factors which were studied were the cropping regime at 2 levels, whereby we had tomato, which was intercropped.
5 minutes.
As a monocrop. The other one is fresh manure at 3 levels, whereby we use cow dung and the goat dung, and the other one is where we had no manure. So you find that the traps, we used traps which we added sterilized soil and we are placed on a clear potting sheet to prevent the nematode from penetrating to the land to avoid contamination. And you find that the slender leaf seeds, which were used as for intercrop, were planted in the sterilized soil at the same time when the tomato seeds were sown in the nursery bed. And find that 2 weeks after transplanting, the second juvenile stage of melanthropic species inoculum was added uniformly in all experimental units, and also at the same time fresh manure was applied in furrows adjacent to the tomato plant. Next. So data was collected on nematode infestation, whereby nematode population was determined in the soil, whereby we took 100 cubic centimeter of soil and determined the number of nematodes. The other one was the number of root guards on the tomato roots, and also data was also collected on other variables like plant height, column diameter, number of branches, and the fruit yield. Next. So data was subjected to analysis of variance using PROC-GLM of SAS program, and those means which were significant were separated using 2 keys honestly significant difference test at P is equal to or less than 0.05. Next.
You have 2 minutes.
So on the result part, you find that the first one is a table showing the effect of fresh crop regime and fresh manure on root knot nematode population, whereby where we had no the crop or where we had no the manure or cropping regime, the population of the nematode were high, while those with goat and the cow and the slender leaf, the population was low. The next slide. The other is on root gall. We also find that the same case occurred where we had no manure or cropping regime, the number of root galls were lower compared to those with cow or goat dung or the slender leaf. The next one. On the growth part, we find that although the plant height were not significant in all the treatment, but those with these 2 factors, where the 2 factors were used, the plant height was at least higher than where there was no manure or the slender leaf in the crop. The next one. So the case also applies to the column diameter, whereby where we had no manure, the plant column diameter was smaller compared to where we had the either crop, and also where we have the fresh manure. Now, so on the number of branches, and we find that where we had the two factors system, number of branches were higher, and you know the number of branches are yield component, whereby if you have more branches, it means that the bearing service for the fruits will be more, where we have more branches. Another one. Next. So this one is the table shows the effect of the cropping regime and fresh manure on fruit, whereby the first section we have the number of fruits. So you find that where we had goat manure, the crop is a slenderly and the highest number of fruits. And where we had no manure nor the crop, the slenderly, the number of fruits were lower. Also the same case applying to fruit weight. And you find that those treatments which had better plant growth resulted to better yield, and also those treatments which had lower number of juvenile population of root node nematodes and also gas and the higher yield. So it shows that these root node nematodes affect the plant growth and yield. Next one.
Okay, wind up, please.
As a conclusion, the result of this event demonstrates how use of fresh manure and cleaner dips can be used as a viable alternative method for management of root nematodes to the result an increase in growth and yield. And we find that this study also forms a basis for advocating the use of fresh manure clearly as an alternative effect method for draw nematodes in often field tomato growth. And we find that combined use of goat manure and slender leaf resulted to the best management of root node nematode and also higher yield. Thank you.
Thank you very much, Munyoki. You have one question that you can respond to.
Two. There are two questions on
Okay.
the chat. You can respond to them. Okay, let me read them out for you. One, what is the concentration or the number of nematodes that were used in the inoculation in your experiment? And two, after how long was the manure used after defecation by the animal?
Okay. Oh, Munyoki? Have you disconnected? We did. Oh. How come? Okay, there is back. Okay, Munyoki,
Recording in progress.
Hello? Yes?
please respond to those two questions. Did you get them?
Yeah. So the number of, the first question is the concentration number of nematodes used. I used 250 nematodes, given a nematode, which there was a procedure of extracting the nematode. So I extracted the nematode and then I planted them 2 weeks after the tomato transplanted. So there are 250 number of nematodes. Because initially I had sterilized the soil. The other question is after how long was the manure used after defecation by the animal? The manure was fresh manure, so fresh manure sometimes affects the plant. That's why I make the furrows just adjacent to the plant. It was not in contact with the plant because during its composition may affect the plant. So I made furrows near the plant. Thank you.
Okay. Thank you very much for that presentation. We'll still engage on the chat so that you can respond to some of the questions that have not been asked directly. Thank you. Okay, our next presenter is Salano. Salano, are you in? And Salano has authored this article with Professor Mulwa and Professor Obonyo. Salano, are you in?
Yes, I am.
Okay, welcome to make your presentation. Please mind the time, so don't spend too much time on introduction. give us the key elements of your research and the findings and recommendations. You have 10 minutes for this and 2-3 minutes for question time. Welcome.
Are you able to see my screen?
Yes, we can see it.
Okay. So good morning. I'm going to present part of my work, and the title is Peanut Accessions Express Low Accumulation of Aflatoxin upon infection. Implications for aflatoxin mitigation. So I'm Elsie Nyangweso Salano, and I co-authored this work with Professor Richard Mulo and Professor Meshak Obonyo. So peanut is a high-value crop nutritionally and economically, and it's ranked among the 5th most important oilseed crop in the world. However, we have one major problem that is related with peanut farming, and that is aflatoxin contamination of peanuts, which is a major food safety issue in Kenya. We've had instances where peanut products such as peanut butter have been withdrawn from supermarket shelves because of containing high levels of aflatoxin. So aflatoxin is a dangerous mycotoxin because it's associated with different health effects such as acute liver failure, liver cancer, stunted growth in children, as well as teratogenicity. So peanut is usually contaminated with aflatoxin during cultivation, that is pre-harvest in the farm, as well as post-harvest. And the contaminated products usually result in economic losses to farmers who rely on peanut for subsistence. So one promising strategy to combat the issue of aflatoxin in peanut is cultivation of peanut varieties with the ability to resist the accumulation of aflatoxins. So the objective of this study was to determine kernel resistance to aflatoxin accumulation in various peanut accessions. So for the materials and methods, 6 main steps were conducted. The first step was sampling. So in sampling, 25 peanut accessions were collected from Kenya, Uganda, and Rwanda. And the reason for this is because peanut is a cross-border crop, and varieties or accessions found in these neighboring countries tend to circulate within the countries. So after that, the accessions were grown under standard conditions because they had been collected from different areas, so it was necessary to grow them under standard conditions and subject them to similar conditions in the farm. The different varieties were harvested at respective maturity dates, shelled, and then sent to the next step of analysis. ELISA was done to measure the total aflatoxin levels, that is the pre-harvest levels. At this point, inoculation was done with toxigenic Aspergillus flavus species, and the samples were incubated for 7 days at 28 degrees. After that, the infected kernels were then subjected to another ELISA test to measure the levels of accumulated aflatoxins. Thereafter, data analysis was conducted, and paired t-tests were done to compare the aflatoxin levels before and after inoculation for each variety. So the experiments were done in triplicates, and the data analysis showed that inoculation of peanut with toxigenic Aspergillus flavus led to an increase in total aflatoxin levels, at least in most of the varieties. So during the analysis, accessions that had significantly lower levels of aflatoxins before and after inoculations were classified as resistant, whereas those that the differences, or those that accumulated more aflatoxins after inoculation were classified as susceptible. So in total, out of the 25 accessions that were tested, 64% exhibited kernel resistance to aflatoxin accumulation, whereas the rest were susceptible. 2 out of the resistant accessions exhibited a very unique phenomenon in that instead of them having high levels of aflatoxins after inoculation, the levels tended to reduce, and this was a unique phenomenon that is yet to be reported. So this is a graphical representation of the peanut accessions and the outcomes that were shown. So out of the 25, more of them, 64% were resistant, whereas the rest were susceptible to aflatoxin accumulation. So the conclusions that can be made from my study is that different peanut accessions respond differently to biotic stresses such as colonization with Aspergillus flavus and the subsequent accumulation of aflatoxins. So the accessions in the unique phenomenon pointed towards novel peanut accessions that can be exploited further in the breeding of peanut for aflatoxin resistance. A few recommendations were made from the work that further studies should be done to determine the genetic source of resistance in the various accessions. Then we can also encourage farmers to grow some of these aflatoxin-resistant accessions in a bid to combat the problem of aflatoxin contamination of peanuts. Then we can also conduct more studies to find desirable traits such as yield, because you may find that there is a, I didn't do this in my work, but there is a possibility that accessions that are resistant to
the numbers,
to aflatoxins are not high yielding or do not have other desirable traits. So there is a need for additional studies to check more desirable traits and merge them in breeding studies to develop new varieties. So I would like to acknowledge Egerton University for providing the facilities to conduct this work, particularly the mycotoxin research lab at the biochemistry department. I would also like to acknowledge the African Development Bank for funding my studies and the research work, as well as the National Research Fund. Thank you.
Thank you very much.
Yes, we have 3 questions. One is, what is the percentage loss caused by aflatoxins in Kenya? The next question is, what data is available on the health issues caused by aflatoxins? And the third question is, could there be accessions which are resistant to A. flavus and A. parasiticus? You can respond to them, Salana.
I'll start with the last question. Yes, there could be accessions that are resistant to F. flavus. As was seen in my study, I inoculated them with the Aspergillus flavus and some were resistant, but I did not use Aspergillus parasiticus, so I cannot say with certainty regarding Aspergillus parasiticus.
Next.
And then data on health issues caused by aflatoxins. Yes, there is some data on that. A number of studies have been done, and the aflatoxins are linked to different crops because aflatoxin does not only affect peanut, it affects other crops such as maize and sorghum. And so the data is diverse based on the particular crop that was investigated. So a recent study has shown that sorghum, contamination of aflatoxin in sorghum has led to malnourishment and stunted growth in children in Kerio Valley. Yeah, so the data is available, but on based on different crops that have been studied. Then the first question, the percentage loss caused by aflatoxins in Kenya. I do not have those facts, but what I know is the economic loss is estimated at $750 million worldwide. But for Kenya, I do not have those statistics at hand. Thank you.
Thank you. Okay, thank you for that presentation, Salano.
Yes.
We want to move to our next presenter, and that is Professor Mungai and Korir. I'm not sure whether Korir is in or it's Professor Mungai who will present.
Hello.
Okay.
Yeah, this is Korir.
Okay.
Yeah, I'll present on that paper.
Mm-hmm.
Yeah, so let me join in through my laptop quickly.
All right.
Okay.
10 minutes, key facts, key findings of your research. What can we take away? So please minimize any other extraneous material.
Okay.
Okay.
Just give me a minute.
Korir, are you ready? Or we can slot you for the next presentation?
Maybe I can go to the next. Maybe you can move to the. I'm trying to connect it. It's giving some problem, but I think you can go to the next and then I'll come after.
Okay.
Okay.
The next presentation would be Kipngetich. Kipngetich, are you in? And are you ready?
I'm ready.
Welcome then.
Thank you. I'm waiting for the slides, so can I share?
Yes, please share. You can share.
So my name is Shadrach. I'm present on suitability, production, and utilization of coffee in selected counties of Kenya. My co-authors are Nancy Mungai, Anadu Pio, Professor Mirionga, and Ezekiel Korir. So coffee is a drought-tolerant crop, and it can offers an effort of combating nutrition, environmental degradation, poverty, and food security in sub-Saharan Africa. In Kenya, it is the second most important after legume. Despite its importance, its production has been low. Coffee plays a very essential role as a food security crop. And despite its importance, yield has been low. The actual yield has been low. The actual yield is 0.8 tons against the potential yield of 1.8 megatons per hectare. Lack of proper assessment of land suitability could attribute this and inadequate information on utilization. The objective of this study was to improve food security through the adoption of drought-tolerant crops, that is cowpea, and specific objective is to develop suitability map for cowpea production, determine the production trends of cowpea in selected counties in Kenya, and provide insight on utilization. Methodology, determination of cowpea production in selected counties. A questionnaire was developed where the key interest was to look on the areas under cowpea production in hectares, cowpea production megatons, which is equivalent to 1 ton, production in megatons per hectare, and the arable land under cowpea production. Additional data was sourced from Kenya National Bureau of Standards. Both data was harmonized and analyzed using Excel.
Recording stopped.
Under the objective 2, development of suitability map for cowpea production in selected counties, data on favorable soil temperature, rainfall evaluation was gathered to develop land suitability. Data ratings to those factors was reclassified in 5 classes: is highly suitable, suitable, moderately suitable, marginally suitable, and currently unsuitable. This criteria was established, was then applied to GIS to determine the different level of land suitability for the cultivation of cowpea. For the objective 3 on the utilization of cowpea, a comprehensive literature review is done using Google Scholar and focused on the papers published between 2004 to 2020. The information on utilization was then extracted. And the results is the cowpea
Recording in progress.
production in the selected counties. This shows that cowpea production is below the potential yield in most of the counties, with Kajiado County having the highest in 2013. Actually, it supersedes the potential yield, and this will be explained on the land suitability maps. In terms of areas under cowpea in the selected counties, the production in these counties is low, with Kitui and Kajiado and Kisumu counties leading in production, despite the fact that the production in these counties is low. The land suitability was found out that Kajiado County was the leading in terms of area suitable for cowpea production at 78%, followed by Kitui at 44%, followed by Kitui at 42%. It was then followed by Nakuru County at 42% still, and Bomet County was the last with 15%. On cowpea utilization, cowpea is utilized in various ways, where the leaves are utilized by leaves are utilized, grains, the green grains, the hulms from the cowpea is used as fodder. It also plays a very essential role in improving soil fertility through nitrogen fixation. One of the health issues that is associated with cowpea is it contains antioxidants, which is able to combat, to remove the free radicals, which is associated with a reduction in terms of hypertension, cancer, and stroke. In conclusion, cowpea production in selected counties is low. Therefore, there is the need to disseminate information on land suitability for improved cowpea production. In terms of utilization plays a very essential role in health and soil fertility improvement, and hence the need to adapt to cowpea. So let us all cultivate and utilize cowpea for improved nutrition and enhanced food security. Thank you very much. I want to acknowledge Egerton University for organizing this conference, and to Forum through the GLDC for funding this project. Thank you very much.
Thank you for that presentation. Do we have any questions on the chat box?
Yes, there's one question which is asking if the areas surveyed already grow cowpea, what is the justification of the study?
the areas we selected was basically those that grow cowpeas like Kitui, Machakos, and Homa Bay. But we found out that other counties like Bomet County, they are not producing the coffee production. And this study was based on use of availability. We selected some students to go and collect data in these counties. So it was based on the ease of access to these, the source of information from this county.
Okay, thank you very much for that very brief presentation and keeping time. Korir, are you ready? Or not yet? Seems he's out. We'd like to move on and welcome the next presenter, and that is Obondo, who is supposed to give us a talk on effects of water hyacinth compost on soil properties and maize growth and yield along Lake Victoria Basin. Mr. Obondo, you ready? Please check for me. Mr. Obondo? Hello?
Hello, Prof.
Can you hear us?
Yeah, this
Hello.
is
Is Obondo in? Okay, our next presenter is supposed to be Mr. Obondo. Is Obondo in? Professor Munga, Professor Lelei?
Hello, Prof. Can I interject?
Okay.
Yeah, this is a Korir. I've been able to set up my computer.
Mm-hmm.
I don't know if I can go next.
You're able?
Yes, I'm able to. Okay.
Okay. Can you
Okay,
proceed then, please?
Okay, thank you. So let me share the screen. So for this, we are doing an assessment. The title is Assessing the Efficacy of Commercial Phosphorus Products for Improving Maize Production in 2 regions of Kenya. So this work was done by Professor Mungai and me, Korir Hezekiah. And so as an introduction, know that maize is a staple food crop in Kenya. And we see that about 1.5 million hectares are planted annually with this crop. And majority of these are, 80% of them are smallholder farmers. But we have seen that over the years, the yields have been going down. And this is because of the low soil fertility. And we see that over the years, people have been increasing the production of, or they have been increasing the amount of fertilizer that they use in their farm, but it has not been going hand in hand with what they produce at the end of the season. And so what we need to do, we need to put in more nutrients in the soil, and these nutrients must be those that are cheap or low cost nutrient fertilizer, and also have some soil conditioners in them, and also have those that are able to boost the microorganisms that can mobilize these nutrients. And because of this, within the market, the agrovets, we see that there are a lot of biological products and foliar fertilizers. And when we have a lot of this, they raise a lot of quality questions to the farmers in the end. So there's a need to have an appropriate mechanism to see if these products meet the claims that they have. So we had two objectives. One was to evaluate how effective these commercial non-conventional products were, and then to determine the efficacy of these products when we apply together with either the rock phosphate or the triple super phosphate that we found in the market. So this experiment was done in two sites, one in Chuka and another site was in Egerton. So these are two different soils, and we selected this so that we have to see how will this product do in a wide range of conditions. So for the two different sites we use, for Egerton we use the hybrid 614 because it's in that environment. And then for Chuka we use the Duma 49, and then this was done in 2 cropping seasons. So these are the products. We have the Symbion P, which is it has some phosphate solubilizing organisms. The Symbion Vam is a mycorrhiza organism, and then the Digro is a foliar micronutrient product. And so these products were applied as the manufacturer's recommendation on the label. So we had treatments where we either had a sole application of this treatment or we combined them either with rock phosphate or triple super phosphate, either at half or full rate of each. And then the full rate of the application, at least we wanted to supply 50 kgs of P per hectare. And then the treatment were arranged in a randomized complete block design, and then you were able to take that on the plant height, the biomass, and the yield. And also at the end, we did an economic analysis, the benefit cost ratio. So on the results, so this is the effect on the plant height of these products. So we did it at 4 weeks after planting, 6 weeks after planting, and also at 9 weeks after planting. So you see at later stages, at 6 weeks and 9 weeks after planting, the control was significantly lower in height compared to the products, meaning that the products were able to boost the growth of the maize. The same with when we compare the 2 seasons, we find that in both the 2 seasons, especially the first season, the control was yielding significantly less, or the height was significantly less for the control. So this is for the biomass. And now we're testing the P sources. When we look at the rock phosphate, the TSP, and when we combine them, and now we try and see how do they interact with our products. So we find that especially when it comes to rock phosphate, there was a positive interaction with the Symbion P. And when it came to TSP, so that TSP was doing well when it was combined with Digro. This is for the 10 weeks after planting. And then also at harvest we did, at planting we also took some biomass. And the trend is similar. When we combine with the other products, they yield more biomass. And lastly, we also did on harvest, the grain yield of the maize. And the first graph is on the nitisol that is in Chuka. So we find that when you compare the control and the application of these products, we see that these products is able to increase the grain biomass, the grain weight of maize. This other, the B part of the chart is at Egerton. So we see clearly that, for example, during season 1, the control yielded much less than the application of these products. And especially when we look at the Symbion P yielding more maize than the other. The same in the second season. We find that during the second season, the same was true, showing that when we apply these products, it's able to increase the yields of the maize. So we did an BCR, the benefit cost ratio, seeing at how much is being spent and how much yield we are getting. And we found that for this product, the Symbion P, when it is applied alone with no other sources of phosphorus, in 2014, it was giving a BCR of 23.4, and in the second year, 18.4. And the cutoff for this, it should be around 2. For the benefit cost ratio, it must be above 2 to show that this practice is profitable. So you see for the Symbion P, it's higher than compared to the others. So when we come down here, like the dry grow, we see they even have a BCR of less than 1. That's for the Egerton site. And then when we look again at the Chuka site, the nitty source, the same is true. The Symbion P without any other addition for the first it was 3.2, and since you are using the same field, in the second year it shot up to 16.69 as compared to others that yielded more benefit as compared to the costs. So in conclusion, you see that these biofertilizers that contain the phosphate solubilizing organisms, they have a great potential for being used for sustainable crops, along with when we optimize how much P we are applying. And so when we inoculate with the P solubilizing organisms, this can be a very good, promising technique because it will be able to increase the availability of phosphorus in the soil. And again, when we apply the rock phosphate, which is an organic source of P, when we combine this with the P solubilizing organisms, it becomes more effective, especially in the acidic soils of Chuka. And again, Digro is a foliar fertilizer. It can also be used, especially when we have issues with P fixation in the soil. This can be used as a foliar fertilizer to supply the nutrients to the crop. So as a recommendation, these products can be used. They are promising that can be taken up by the farmers. So thank you. This study was supported by the Bill and Melinda Gates Foundation under a project through IITA called the COMPRO project. Thank you.
Thank you very much, Korir, for your very illuminating presentation. And we
have about 4 questions.
Okay.
But
you, for time, I think you respond just to 2, then the rest you can respond later.
Okay, and also Kipngetich can respond to the questions that are posted on the chat, the ones he did not respond to. So the first question, Coril, is did you consider the farmer's practice, for example, the use of DAP in your experiment, and were your crops top dressed? The next question is, are the soil amendments, the ones that you used, were they analyzed for residues to know their purity? And did you analyze to check and confirm the nutrient specified by the manufacturers, because they are business people, they may be doing business? Thank you. You can choose the ones to respond to.
Okay, thank you. So the first question on the farmer's practice, yes, we have the positive control, whereby now after we planted with the TSP, and then we came and top dressed with an equivalent amount of nitrogen during the top dressing to show how the farmers do their thing. So the farmer's practice and the top dressing was in the positive control. And when it comes to the analysis of these products, so 2 of the products, these are the biofertilizers. So what we did, we did a lab study where we checked on the purity of those organisms, where we did some lab analysis and checked on the purity of both the Symbion P and also the mycorrhiza product. Okay, thank you.
Oh, there's still time? Okay. There are 2 other questions which you may respond to. I think we have like 2 minutes for that.
Okay.
You can respond to the one on the nutrients, whether they match the ones which are specified.
Yes, so for the digro, so for the two, the Symbion P and the Symbion, yes, we did some lab studies. We cultured them and we saw on their purity if there are any contaminating organisms. And we saw that both the two biofertilizers were up to the label. And also for digro, we also did similar, and then we checked at the nutrients levels within the products.
All right, thank you very much for that information. Our next presentation is from Mr. Obondo.
Yes, can you hear me?
Yes, we can. Are you okay?
Just a minute. Just a minute. Okay.
Excuse me. Oh, it's gone off. Oh,
Recording in
Type
progress.
of compost and the quality depends much more on the method.
hello?
Goes
Hello?
with
Maybe you need to
maturity.
restart.
We cannot see your screen.
We were testing the three system of composting. One of them was vermicomposting.
Hello?
The other one was EM composting, that is composting using effective
Hello?
organism. Yes.
We are not able to see your work, and you need to go back a bit, rewind. Start afresh, please. We'll give you extra minute.
Yes, moderator, I think he needs to open up his slides, eh?
Yeah.
Because what we are seeing is just his screen, eh?
Oh, okay. Sorry. Let me work on it.
The other presenters, kindly share your PowerPoint so that we can share from this end.
Ele Ouma.
Yes,
You are ready?
yes, I'm ready.
Are your slides available?
Yes.
Okay. Ubondo, do you need the extra time, please?
Just give me extra time. I work on it kindly.
Okay. So we'd welcome Ouma.
Okay.
She's?
Kobondo, please stop sharing your screen.
Yeah. Okay, Lillian, you are on.
Okay. I'm sharing the screen. Are you able to see my screen?
Okay, put it on presentation mode.
Okay. My name is Lilian Ouma. I'm sharing a paper on lignocellulose component of potential fodder sorghum genotypes as influenced by genotype, stage and environment, and I co-authored this paper together with Professor Cheriot and Professor Gendo. So sorghum is a versatile and drought-tolerant crop, which is cultivated in most tropical and subtropical countries of the world. It is ranked 5th among the cereals, with an approximate production of 65.53 million metric tons. And Kenya is ranked 6th in sorghum production in sub-Saharan Africa. Sorghum as a crop has a rich genetic diversity and hence has multiple products for both domestic and industrial uses, for human food, industrial use, and also for biofuel and alcoholic beverages, as well as it is used as livestock feed.
Okay, please move your slides. You're still on slide 1.
From my side, it is moving.
Wow. Can you help us share?
I've given the authority for quality control.
Okay, just proceed as they sort it out.
Okay. I was in the introduction, and I was saying that the livestock sector in Kenya has been increasing, and therefore this has increased the demand for feed. However, the gap between the feed demand and supply is still wide. Feed deficit in Kenya is estimated to be as much as 70% of the total annual fodder requirement. The feed sector in Kenya faces different challenges, such as low feed quality, inadequate supply of feed, overdependence on maize for livestock feed, and environmental issues like drought, pests, and diseases. So normally the livestock sector is dependent on maize, as I said, for feed, but maize has several challenges, and sorghum can be a good alternative for fodder source. However, sorghum also, can you hear me?
Yes, we can hear you. Proceed.
Okay. Sorghum also has a challenge of high lignocellulose accumulation during crop growth period, and lignocellulose reduces digestibility of fodder in the rumen. And therefore, the objective of this study was to determine the effect of environment and stage of development on lignocellulose component of selected potential fodder sorghum genotypes. I think now it's being controlled from the center. Next slide. Materials and methods. The experiment was conducted between March and September, the growing season in 2019, and repeated between May and October in 2020 in 3 locations with variations in temperature. That is Egerton University, which is at AZ3, Rongai, which is at agroecological zone 4, and in Marigat at AZ5. The plant materials were sourced from ECRISAT, GeneBank, and from farmer selections. The sorghum genotypes were evaluated in a field experiment in a RCPD design with 3 blocks and 20 treatments, which were the 20 genotypes. Those were the specifications of the experiment. Then in 2020, 10 sorghum genotypes, which were selected subjectively from the experiment in 2019, were planted in a separate experiment to aid in measurement of plant growth parameters. Growth parameters that were measured were plant height, days to 50% heading, and we also measured average building temperature. For sampling, the sorghum was sampled at 2 developmental stages, as shown in the picture, at booting stage and at dust stage. And for each development stage, random sampling was done at the 3 middle rows in each experimental unit. Then the samples were shredded and taken to the laboratory for evaluation of lignocellulose content using Van-Soest method 1991. Then data was analyzed using analysis of variance was done using in GNM procedure in SAS, then mean separation done using LSD, and then correlation analysis was done using Pearson's correlation coefficient. The results, days to 50% heading. This was affected by both significantly affected by both genotype and environment. As we can see, the different genotypes, 10 genotypes that were selected at different heights, and the plants that were grown in Marigat were shorter compared to Egerton, while those in Rongai were intermediate. Plant height also varied among the genotype, and genotype E35, the shorter plants, the shortest plant, while genotype E6518, which was our control, this is a fodder genotype by Kenya Seed, had the tallest plants. And again, plants in Marigat were taller in most of the genotypes compared to Egerton and Rongai. The lignocellulose content, the results indicate variation in lignocellulose content across environment, and there was also a significant change in lignocellulose content as crops matured. This table here shows the cellulose content at booting stage and at duff stage in the year 2019. In 2019, we grew crops in Egerton and Marigat only. And as we can see here, B35 genotype, which is a staging genotype, had the lowest cellulose content. EST 37, which is a PMR trait, and D6518 also had lower cellulose content. The ones highlighted in red, that is IS 1116 and the other two, had high lignocellulose content. Our control was this E6518, which is a fodder genotype. So compared to it, these 3 genotypes highlighted in blue had lower cellulose content. The same trend was observed in 2020 as cellulose content increased as we moved from Egerton, Rongai, to a hotter region in Marigat. Again, different genotypes in 2020 had different cellulose content, with B35 having the lowest, and the ones highlighted in red having highest. The cellulose content followed the same trend as cellulose. Again, B35, EST37, and E6518 had lower cellulose content, while these highlighted in red had higher hemicellulose content. And again, environment affected hemicellulose content with crops that were, with plants that were
1 minute.
Okay, sorry, 1 minute.
1 minute.
Okay, lignin followed the same trend. And then the correlation, this is a table showing the correlation between lignin, days to 50% heading and plant height. And as you can see, the coefficient of determination, the percentages, indicate that variations in lignin content may be attributed to differences in days to 50% heading and plant height. The discussion, variation in lignocellulose content among sorghum genotypes could be attributed to differences in plant architecture and also differences in the genetic makeup of the plants. EST37 is a BMR trait which is associated to low lignin content. B35 is a stegrine and a dwarf sorghum which took shorter period in the field to achieve maturity, and therefore lignin accumulation did not take longer period as compared to the late maturing genotypes. Increase in lignocellulose as plants increased in maturity could be attributed to buildup of secondary plant cell walls which provide strength as plants matured. Conclusion, genotypes B35, EST20, and EST37 ranked low in lignin, cellulose, and hemicellulose content, and soda fodder harvested at rooting stage had lower levels of lignin, cellulose, and hemicellulose. We therefore recommend that based on lignocellulose content, the best stage to harvest fodder sorghum to achieve high fodder quality and higher digestibility is rooting stage. And we also recommend that in vitro and in vivo digestibility analysis to be conducted for the highlighted genotypes which had low lignin content, that is B35, EST20, and EST37. The authors acknowledge some for financial support towards this research. Thank you.
Okay, thank you very much, Lilian Ouma, for that presentation. Any questions on the chat box?
Yes, there's one question on the chat, and it's asking, why did you determine the lignocellulose content at dose stage, which is beyond the fodder stage? You can respond to that.
Okay, the recommended fodder stage is normally between rooting stage and dwarf stage, somewhere around the bloom stage. But also, as you check, as you consider lignin content, biomass is also a factor. Biomass is also a factor, a quality factor of fodder. So if you harvest a bit earlier, this may have an implication on biomass. So the reason why I went up to dwarf stage is because in my other objective, I was also looking at the biomass content of fodder. So you realize that when you go up to dwarf stage, the biomass is higher, even though the lignin is a bit, the lignin content is also increased at that level. So it's a compromise between lignin content and biomass.
Okay, if I may ask a little bit also on that, was it hard dose stage or soft dose stage?
It was somewhere between hard duff and soft duff, a stage where the grains had a duff consistency when pressed between fingers, so it was somehow soft. Not the milky stage and not the hard duff.
Okay.
Thank you. Anybody else with a comment or a question can post them on the chat and Lilian can respond later.
All right. Thank you, Lilian, for that illuminating presentation also. And we want to move on. I don't know if Chebet is ready. Chebet, Njogu, we have 2 more presentations to go before our break time 3, actually. Ubondo has not presented, but we'll take whoever is ready.
Ubondo is ready.
Okay.
Yes.
Please.
Able to see my screen now?
Please put on your slide mode so that we can see your slides.
I'm sorry, my presentation in the form of a PDF.
Whoa.
I did not prepare the slides, but I'll just go through very quickly.
Okay.
Okay, sorry. So, thank you once more My name is Obondo Kevin, and my co-authors are Prof. Samuel Mwonga and Prof. Joyce Ndemo, and we're working on the effect of water hyacinth compost on soil properties, maize growth and yield in Lake Victoria Basin. We did our experiment in 2 sites, that was in Asembo, Siaya, and in Kisii. Experiment 1 was done in Siaya and experiment 2 in Kisii. So crop production in many parts of Kenya become uneconomical without the use of fertilizer, but because of the high cost of chemical fertilizer, the application rates are very low among many farmers than the recommended rates. And compost is one of the organic amendment which is also rich in essential plant nutrients, which can be used to improve crop yield. Composting can be achieved through 3 main processes. That is composting using using the redworms, or what we call the earthworms, which are specialized in degrading organic matter. We can also do it using what we refer to as effective microorganisms. These are microorganisms which also aid in accelerating the decomposition process. It can also be done by use of the usual way which we are used to, the thermophilic system, which relies on the organisms in their natural setting. Okay. So when we talk about composting, we have a problem with access to organic residues, because some of them, like the crop residues, we use them also as livestock feeds, and also other activities, and therefore getting them must become a problem. And so we thought about using water hyacinth as a substitute. But again, now when you're talking about use of water hyacinth, which has been a menace in the Lake Victoria region, our aim was to find a way in which we can improve its quality, so that even when we say you use compost manure as an alternative to chemical fertilizer, we can be able to get maximum yield by using a quality material. That one means you will use lower rates compared to the normal when we do the thermophilic system. So briefly on our methodology, how the composting was done, we used beds, then we replicated the beds 3 times. We used 3 beds for the 3 systems, replicated 3 times. So the picture there shows the manual process of harvesting the water hyacinth from the lake and how it is dried before it's taken into those 3 systems. More detail can be found on our on the official website, where we had the full presentation. So at the end of the composting, I will just be brief. At the end of the composting, we decided now to do an experiment and compare it with now what farmers has been using, which is the chemical fertilizer, and find out the differences in yield, what we can be able to get with the compost and what we have been getting with the chemical fertilizer. And also we had a negative control where we did not apply anything in our field. Okay. So on the results, one of the main interesting result was on the quality of the compost, which is also one of our main objectives. And we realized that when compost was the best. We realized that when the composting was the best method in comparison to those other methods. And we also realized that this water hyacinth, it has a very high concentration of these essential plant nutrients. Most of the 3 primary macronutrients, led by nitrogen, we have phosphorus, and also we had potassium, being very high in vermicompost compared to these other composting systems. Another potential result was on the maize growth parameter. Trying to check and in the photo here you can see the plate is in one of our experimental sites where we were comparing the different fertilizer types. And at the center you can see here is where the maize there are very dwarf. And at the end here is where we applied the chemical fertilizer, and on the other end is where we applied the one of the organic amendment. Of course, this one was vermicompost, and we had others also within the system. Okay, so again, on our parts of result, one of the other interesting bit, especially to the farmers, so now in terms of the grain yield, when we talk about maize as a test crop, and we realize that with maize, with vermicompost, to get the highest grain yield in both sides, one site in Siaya and the other site in Kisii, compared to the rest of the treatment. And then this one was followed closely by the chemical fertilizer, in this case in Siaya, then of course the other compost types. Of course, the negative control, where we didn't apply anything, we got the minimum yield. Okay, approximately 1 ton. Well, where we were using vermicompost, we got 4.5 times. So it was 4.5 times where you don't use any fertilizer, and at least 10% higher than when you use chemical fertilizer. And also on soil properties, okay, we can check on the right flank. Initially, our soil in Siaya was moderately acidic, a pH of 5.2, and when we applied these organic amendments plus the chemical fertilizers, these were some of the changes that we were able to get in our system, in our field. Okay, for example, where we applied vermicompost, the pH managed to rise from 5.2 to 5.7, and where we used TM compost, it was a slight change by 0.1 unit, the same as for thermophilic compost. Where we used inorganic fertilizer, the chemical fertilizer, the pH dropped by 0.4 units from 5.2 to 4.6. And where we used, where we did not use any fertilizer, again there was a 0.4 unit drop in the pH. This one shows that even where we don't use fertilizers, as long as there's precipitation during the growing season, as the plant takes the basic cations, there are chances that what is left behind is the acidic cations, which in turn drops the soil pH. The same applies also to electrical conductivity, though this one was not much affected because it was within the optical range for most of our field crops. So in conclusion, we can, from our findings, we can conclude that the result of the study demonstrate that the best quality compost was obtained with vermicompost, and that the use of water hyacinth as a substrate for composting has shown the potential to improve soil fertility and eventually crop yield. And the long-term adverse effect of chemical fertilizers, we can see, for example, under the result under the effect on the soil properties, how it is reducing the pH. The long-term adverse effect of the chemical fertilizer, especially on soil and environment, makes their use undesirable. Therefore, compost from water hyacinth, which is locally and readily available in the Lake Victoria region, we can make use of it as an alternative to chemical fertilizer, whose price is also becoming unfriendly to most of our farmers. Thank you very much. I would like to appreciate the collaboration between Forum, Egerton, for organizing this conference to enable us to get more insight about our current research and application. Thank you.
Okay, thank you very much, Kevin, for that presentation. I think you have like 2 minutes, 3 minutes to respond to some of the questions that have been asked.
Yes, we have 2 questions. How much dry matter did you obtain from the fresh hyacinth? The next question is, what precautions were taken to restrict the potential risk of spreading the noxious weed? And is it possible to use water hyacinth as green manure? Because it seems a rigorous process to get it. And would the community around the lake adopt this strategy? Thank you. You can respond to 2 and then you engage in the chat.
Okay. So for the first question, what I estimate is around 92% water. That means if I want 1 ton of dry matter, you multiply by around 90%. I need to go for close to 10 tons of wet matter. Okay, to convert it to 1 ton of dry matter, you need to go to about close to 10 tons. So in our case, for the experiment, we harvested 1 ton. Then this one we converted, and we were able to get something close to 100 kg of dry matter for each experiment. And this one we did it 3 times. So we harvested the total material that was used for the composting was 3 tons. And this one was able to give us around 300 kg. The second question, a plant once seen as a noxious weed, a threat to the lake environment, is no longer a noxious weed because it has now found numerous potential uses, one of them being used as compost manure. And so the risk of spreading it to another environment is no longer a big question at hand. Okay.
Hello? I think
Yes.
one more question that you can respond to.
Okay.
Or you've forgotten what it was?
You can check on the
Yes, you want me?
chat. You can check on the chat the ones you have not responded to.
Now it's talking about, is it possible to use water hyacinth as a green manure? Because it seems the process is very laborious.
Okay. Thank you for the reminder. Okay. So in this process, I have not explained it in detail because of time, but I will like to share it later on, the whole process on how it is prepared, and there's also comparison between the green and the composted form. The green form is not very much efficient in terms of the release of the nutrients. The nutrient in the green state is not much bioavailable compared to when it is in the composted state. So that is the main difference, but it is possible you can use it as a green manure. But if you want a quality one, then it is better we go to the process of the compost, which again is not very much tedious and is more friendly, more bio-friendly compared to the green state. Thank you.
Okay, thank you very much for your presentation. That presentation raises a lot of questions scientifically. I do not know what we think about the issue of being a noxious weed. But those are some of the things that one needs to think about as we develop innovative technologies to avoid creating greater environmental problems through the technologies that we create. But we really appreciate this new way of using what was considered a problem, and now we are turning it into a very positive way of improving production of our crops. So thank you again, Obondo. We want to welcome the next presenter, who is Chebet. And Chebet is going to take us a little bit into the lab. I know we have done a lot of field experimentation here. So Chebet, are you ready?
Yes, I'm ready.
All right, you put it up for us then. 10 minutes or less.
Hello.
Come on.
Can you see my slide?
Not yet. Okay. Yeah, we can see it now. All right, come on, take on. Time is running, the clock is running. Oh. Ooh. Okay. Hello? Chebet, do you need help? Hello? Okay, as Chebet reorganizes herself, is Njogu in? Njogu? Njogu, are you in for presenting bioactivity?
I'm in.
Are you ready to present?
Yes, I'm ready.
All right, you take over. 10 minutes for presentation. Yes.
Okay, let me share my screens.
Okay.
Is my screen visible?
Yes, yes.
Can you see my screen? Okay. Good afternoon, everyone. My name is Nancy Jogu. I'm presenting to you a paper entitled Bioactivity of Selected Biopesticide Against Fall Armyworm in sorghum in Kenya. And these are my co-presenter, Kamau, Professor Kimuto, Manyasa, Dr. Nyanga, Dr. Charibu, and Mr. Toet. And that is my my presentation, introduction statement, justification, and my tools and methods, and the rest. And this is my introduction. Sorghum, as it has been mentioned by a previous presenter, it is the fifth important cereal food crop in the world. And it is it's drought tolerant. It has water lodging and saline occurring and tolerance. But apart from its potential, there are limiting biotic factors, that is the pests and diseases. And my concern today is the pest fall armyworm, which was first reported in Africa in 2016 and in Kenya in 2017, and it's causing enormous loss in cereal crops. As of now, there is limited documentation of effective control measures, especially in sorghum. Fall armyworm is a Lepidoptera. It undergoes a complete life cycle, and it has 6 larval stages. So, statement of the problem, as of now, there is a lack of sufficient understanding on the type of pesticides and efficient application, and very limited efficacy trials. Farmers are also applying excessive pesticides, and there is risk of environmental pollution. So my current standard, I'm trying to look at efficacy of selected biopesticides in terms of toxicity, deterrence coefficient, and percent dependence in the laboratory at Egerton University. in short, I'm looking at some biopesticides and looking for their mode of activity in managing the fall armyworm. The justification for this work, to provide scientific basis for their recommendations and formulations and packaging. And it also generates knowledge on IPM alternative strategy. Materials and methods, experimental site will be Egerton University. It has been Egerton University, that is in animal science insect rearing facility. And the experimental design is a total of 6 treatments, 3 replicates which are completely randomized. And these are the chemicals which I evaluated for efficacy. The first one is neem plant, which is in Besidine. This is a commercial product. There is 2 entomopathogens, the ovarian bassiana and Bacillus thuringiensis. Diatomite complex, which is mineral-based. And then there was positive control, which has been recommended, that is radiant, and there is negative control where there was no spray. So I started my work by first doing mass layering for the insect, and to have a uniform colony, I went for the eggs which were arranged in the field. I was lucky to get some which were hatching, and I was able to feed them on fresh sorghum leaves, and I maintained the room temperature at 25 plus or minus 2 degrees centigrade using a heater, and I maintained humidity at 60 to 80% using a humidifier. And the larvae, the neonate grew, and at 4th instant, because of cannibalism, I transferred them to a 3-liter jar, and they were able to pupate for 14 days. After 14 days, so I got the pupa. The pupa were removed and placed in 1-liter jar, which was the emergence cage, which was fastened with a rubber band and a muslin cloth so that the insect don't disappear. The adults were placed on the evaporation cage, which was measuring 30 centimeter by that centimeter, and it is indicated. So for the toxicity study, that was the first efficacy study which I was doing. My main aim was to find out the time it would take the instar levels, that is 2nd, 3rd, 4th, and 5th instar level, to be controlled by the different biopesticides. So I coated my experimental containers with each and every chemical. These containers were 7.5 centimeter high and by 6 centimeter. And then I was able to introduce 10 larvae in each cage before introducing treated fresh leaves. After that, since this is the mortality which I was looking at, I was able to count dead larvae after 24, 48, 72 hours after this setup. This was repeated for the 2nd, 3rd, 4th, and 5th instar larvae. I was able to get the actual mortality, that is, by just getting the dead larvae over the total number, that is 10. And in order to correct for the larvae which can die because of the natural causes, I did the collected mortality as per the Humboldt 1925 by subtracting the percentage, the mortality of the control, which was the non-treated one. Then I also did the anti-feed anti-test, that is how far the chemicals can prevent the insect from feeding. And in this case, I treated sorghum leaf disc, 30 millimeter by 30 millimeter in experimental cage, and I introduced 10 instar larvae on the leaf disc, and I was able to get the amount of food which was fed on. And after that, I was able to get the ones which had anti-feedant test and anti-feedant effects, or making the insect not to feed. So here, on this photo, I'm weighing the material before feeding the insect. And here, down here, below here, I introduced the insect and the treated material, and I weighed again to see the amount of food which the insects had consumed. And that is the formula for the anti-feedant test, which was done after 24 hours. Then I also checked the repellents for the various biopesticides. And in this case, I used a circular flat bottom plastic containers. According to Ogendo et al. 2004, I placed 10 insects in the middle, and then I placed alternate treated and untreated sorghum disc at equidistance from the center, so that the insect now can move in order to get the repellents. And in this case, I used the 10 Insta-larvae because it is one which is normally active. And then the total number of insects settling in the control and the treated leaves were recorded after the first, third, fifth, and 24th hours after the setup. And that is the formula for the percentage repellents. And data analysis was done after transforming the data, and the statistical model fitted was R&D, and I used GenStat.
You have 4 minutes.
Yes.
4 minutes.
So these are the, yes, sir. So the results indicated that there was no significant interaction between biopesticide and Laboinsta. And when I came to but there was significant interaction between biopesticide and time, Insta and time, and biopesticide and Laboinsta and time. When they come to the toxicity, the second Insta larvae was easily controlled, as you can see these figures up here, and also the third Insta larvae, it was easily controlled within the 24 hours. And what can also be noted here is that the fourth Insta larvae were very difficult to control. But also interesting is that the fifth Insta larvae was also easily controlled just within the 24 hours. So for the toxicity, this is clearly seen. The blue line here indicates that the second Insta larvae is easily controlled within 24 hours. And for the biopesticides, second chart, the third Insta larvae is also well controlled within the 24 hours, that is 24. But for diatomite, it only worked after 48 hours. That is where you have the range. And this one is also for the fourth Insta. It was very difficult to control, as you can see. For the fifth Insta larvae, it was very easy to control, in fact, within the first 24 hours. And for deterrence, coefficient, Biavarium Bassiana was the least, while Radiant and Neem added the highest deterrence coefficient. For the repellents, Neem added the highest, followed by diatomites and Biavarium Bassiana. And for the toxicity, for discussions, I've just mentioned that with the 24 hours, 48 hours, 72 hours influenced the larval mortality. And for these, Insta larvae were difficult to control with these bioentomopathogens, and it was easier to control the fifth Insta larvae within 24 hours. And there was significant variability for deterrence and feeding deterrence coefficient for Radiant, followed by Neem, and Pambulant reported the same result. And Neem had the highest deterrence. It has also been reported by other scientists. And this one probably showed that Neem could also repel the insect even in the field. And the conclusion is that the high toxicity antipedant repellence effect by Neem on the fowl larvae shows is a potential biopesticide. And the repellency toxicity effect by diatomite can manage young fowl larvae. And at some specific larval stage and exposure time and biopesticide, each biopesticide is effective within suitable environmental conditions. So for recommendation, I would recommend that Neem, diatomite, Biavarium Bassiana, bacillus can be considered in IPM. But the more field evaluations on efficacy of the biopesticides should be carried out to validate their effectiveness in the field. And also evaluation of optimal biopesticide should be done because I was using the commercial rates or another insect. And the most study to determine the effect of biopesticides on non-target organisms and the environment. Acknowledgement, I'm acknowledging CISAM for funding, the CPE for capacity building on insect rearing, and the field staff at research Kiboko and Koibatek. Thank you for listening.
Thank you. Okay, thank you, Nancy, for that presentation, quite detailed. I think you're fortunate you've benefited from a few minutes here and there. So thank you very much for that presentation. Any questions?
There are no questions on the chat, but anybody who has, sorry, there is one. How applicable are these biopesticides to other plants? And then I would say that anybody else with a question can unmute and ask the question.
Thank you.
Nancy, you can respond.
Okay, I can respond. I can say that they are, through literature, the biopesticides are working very well, even in other insects. Like neem has been used to control aphids and other insects in the field. Like diatomite has been used as a post-harvest storage chemical, but this time I decided to try it in the field and it is working. And it is very cheap, it's only 20 shillings per kilogram. So farmers can just dust it and manage the fall armyworm, especially for the young larval stages, instead of using soil and ash. Thank you.
Okay, thank you very much for that presentation and your response.
There's one more.
Oh, there's another one? Okay.
How feasible is the diatomite in dusting large farms?
And availability?
The feasibility, it is possible because there is a dusting equipment which can be used for dusting. Instead of pressing the granules in the funnel like the way I was doing, there is a duster which can be used to produce fine particles. And although there can be a problem of protecting yourself because you may inhale the dust itself, it's possible. I think it's possible in a large scale.
How about the availability of diatomite to many farmers? How available is it? And the cost?
Okay. In the shops, especially in Nakuru, it's available. It is being mined in Kariandusi. There is a diatomite factory just there next to Gilgil.
Yes.
And it has also been commercialized. It is in fact sold as Kensilf. For the commercial product, it is called Kensilf. And it only costs 20 shillings per kilogram. It's very affordable.
Okay. Thank you.
You can also respond to the other questions on the chat. You can type in to the question.
Or you can read for
Thank you.
Okay. Have you gotten what is on the chat? 2 minutes, or you can respond to that later on, because I think your time is up.
The question is does the concentration affect its workability?
Okay, then the diatomite, you normally get it from the shop and use it as it is. It is normally in powder form. So in this case, you don't alter the way it is just mined and into a powder. And it normally injures the body, the skin of the organism. That is how it normally works. But in my experiment, I've discovered that it can be a repellent as well, and it can also be an anti-feedant.
Okay. Thank you very much for those responses. We've learned a little bit new things concerning diatomite. Now, our last presenter for this session is Chebet, who has co-authored an article with Professor Opio and Githendu Githenku. Chebet, are you in now? Are you ready? Chebet? Chebet, can you hear us? She's off, eh? Oh, she's not in, eh? She's not in.
Can you hear me? Hello?
Yes, yes.
Hello? Yeah, we can hear you.
Okay, I'm ready to present.
Okay, you've eaten into 1 minute, so you have 9 minutes for presentation.
Okay, can you hear me now?
Yes, we can.
Okay, can you see my slide?
Yep, we can see that.
Okay.
Yeah, you
So
go ahead.
my name is Winnie Chebet-Wambogo, and my paper is a review paper on rapid multiplication techniques in seed potato production in Kenya, prospects, opportunities, and challenges. On the introduction, potato is the third most important food crop after wheat and rice, that is globally. And in Kenya, potato is the second most important food crop in Kenya after maize. However, despite potato being the second most important food crop, its production is quite low at 9 to 10 tons per hectare. This is as compared to 20 to 40 tons per hectare that is obtained in well-established countries. Some of the major causes of this low yield is a shortage of quality seed, soil fertility constraints, and pest and disease incidences. Insufficient certified seed potato is a major challenge facing the potato production, with the formal sector producing 2%, meeting 2% of the demand, and this leads to around 98% of farmers to depend on informal seed sources, which are usually obtained from positive selection, clean seed, and farm-saved seed. Rapid multiplication techniques have been introduced to increase the amount of seed stock for seed multiplication, which are disease-free. Some of the major rapid multiplication techniques introduced in Kenya, they include tissue culture, aeroponics, and hydroponics. On the conventional seed potato production, it entails the use of soil as a media for seed production. And as you can see in these charts, 98% of farmers, they rely on the conventional seed potato production method, and only 2% rely on the rapid multiplication techniques. Some of the major constraints that are faced with the use of conventional seed potato production is that the first one, it has a very slow multiplication rate, and it can take up to 7 years to produce sufficient seed potato. And this has led to high cost of certified seed potato, which accounts to 40 to 60% of the total potato production cost. The other issue facing conventional seed potato production is that it encourages the buildup of diseases, that is the viruses, fungi, and also the bacteria. And as you can see in this chart, 49% of the disease affecting potato is the bacterial wilt, which is mainly spread by the use of these seeds. This has created the need to adopt to seed multiplication strategies, which will ensure continuous supply of disease-free seed potato. And this led to the introduction of the rapid multiplication techniques. These RMTs, they provide a distinct opportunity to produce seed potato at an enhanced rate in a controlled environment. And currently, 80% of the total mini tubers and basic seed produced in Kenya, they are produced using the rapid multiplication techniques. These are the mini tubers that are obtained from these techniques. And in this other picture here, we have the RMTs that are mostly used in Kenya. We have the hydroponics and the tissue culture. Some of the major positive attributes that have come up by the use of the rapid multiplication techniques, they include the first one is that there has been increase in the number of seed producers in Kenya. That is from around 12 in 2013 and around 27 in 2019. The other positive attribute is that the potato varieties have increased. That is to around 60. Currently, we have more than 60 potato varieties, and the new released potato varieties by the International Potato Center include Unica, Wanjiko, Nyota, Chulu, Lenana, and Congo. The third attribute is that the certified seed potato has increased. That is, currently we have more than 2 million metric tons of seed potato that is produced annually, and this accounts to an income of about 2 million USD. Despite the introduction of RMTs, there has been low adoption, and this has dragged the quantity of certified seed, which is currently at 2%, and this is because of high investment cost, and it requires advanced skills which might not be available for smallholder farmers. The other issue is that there is lack of awareness by farmers on the use of quality seed. And the last one is that these RMTs, they require high maintenance, management, and installation. The first RMT I'm going to discuss is the use of tissue culture. Tissue culture in potato is a promising innovation with the potential to rapidly reduce the time for seed multiplication and at the same time control the diseases. All types of seed multiplication techniques, they start from tissue culture. This is ADC molar tissue culture laboratory, whereby the in vitros are produced. Once the in vitros are produced, the apical rooted cuttings are then produced. And since the introduction of this, that is the production of in vitros and the apical rooted cuttings, the seed availability gap has been reduced. And it has been reported that the use of the apical rooted cuttings and these in vitros can reduce the cost of seed by 300 USD, and this can increase affordability and accessibility to farmers. Tissue culture has also increased mini tuber production in Kenya from around 30,000 to 1 million, that is in a span of 3 years. Major institutions, there are only 4 institutions in Kenya which deal with tissue culture production. That is the ADC Molo, which produces a capacity of around 100,000 plantlets per season. The second one is Genetic Technology International Limited, which produces by order. We have Stockman Rosen Limited in Naivasha. It produces over 200,000 plantlets. And the Tigoni Potato Research Center, which produces 20,000 plantlets per season. However, as we can see, we only have 4 institutions in the whole country that is only producing tissue culture plantlets, and this is a major challenge that is dragging the potato industry. And one of the major reasons is because tissue culture is expensive. It requires high capital investment, labor, and also energy. And also, it requires different sources of nutrients and media formulations, which can be quite expensive. The second RMT is hydroponics, is the use of hydroponic system. Hydroponic system is a soilless production technique which involves growing plants on an aqueous solution of nutrients or an inert non-soil substrate. It has been reported that 1 acre of hydroponic greenhouse can produce the same yield as 10 acres of field-grown produce. It has also been reported that hydroponically produced seed potato can yield up to 200% more than soil-produced seed potato. We have different media that are currently used in production of seed potato. The most common one is these cocoa peat media, and about 70% of seed production in Kenya is produced using cocoa peat. The second one is sand hydroponics, and we have others such as the pumice, the vermiculites, and the peat moss. This is a picture of the hydroponic system. This is at ADC Molo. These are the apical rooted cuttings, and the media used here is cocoa peat mixed with pumice. The last RMT technique is the use of aeroponic system. Aeroponic system, it involves plant cultivation without incorporating soil or substrate culture. Since the introduction of the aeroponic system, it has led to production of more than 500 mini tubers annually in Kenya. And as you can see in this picture, this is a picture of the aeroponic system. And as you can see here, you can do multiple harvesting. This is by you only harvest the seeds that have reached the diameter that you need. And due to this, they can grow up to around 6 months before you completely remove the crop. And due to this multiple harvesting, the number of seed can increase by 100 to 250% as compared to single harvest in soil cultivation. The aeroponic system, it stands out amongst the RMTs used. This is due to production of high number of tubers. However, the adoption rate is quite low, and this is due to high investment cost, and it totally depends on electricity. it has been reported that it has an initial cost of around 240,000 USD, which is required to set an aeroponic system that can produce 80,000 mini tubers annually. In the conclusion and recommendation, the introduction of RMTs in Kenya has facilitated production of disease-free seed potato and has significantly increased the availability of certified seed potato. In my recommendation, an integration of both formal and informal seed system involving the private seed sector, progressive farmers, and cooperatives is required to bridge the demand gap in supplying clean seed material to the farming communities. Also, these production techniques, they need evaluation in terms of productivity, profitability, and sustainability, and also a comparative analysis can be done between the hydroponic and aeroponic system in terms of cost per tuber. I acknowledge Forum and TAGDEF for their support in my academics. Thank you.
Thank you very much, Beth, for your presentation. We have a few questions.
Yes.
That you can respond to.
Yes, there are 3 questions. How are these plantlets produced? How are these plantlets produced resistant or susceptible to biotic factors? 2, what are the cost implications, that is time and money, of using tissue culture in developing potato planting material production to the point where they are viable seed potato? Number 3, why are these centers producing controlled amount of plantlets instead of mass production? That is, the question is not very clear, but James Othier can ask. You can respond.
You can respond, and any of your co-authors, if they are in, they could also respond.
Yeah, can I respond that in the chat?
Yes, please.
Okay. Thank you very much.
James, you wanted to ask a question?
Yes, I wanted to ask a question. Thank you. I'm asking why the centers produce controlled amount of plantlets, but not mass production, because the area coverage and the farmers, they're saying that they produce less compared to the productivity.
Yeah, the answer is because these tissue culture labs, they have the capacity and they cannot surpass their capacity. And also, so like according to, and also because of the seasonality of the potato, sometimes they cannot produce quite huge numbers of the plantlets, and yet it's not the season that is not the season for planting the potato.
Thank you.
Thank you. Chebet, you can respond to the cost implication of doing tissue culture.
Chebet, you got that question?
Sorry, I didn't get the question.
What are the cost implications, time and money, of the tissue culture in potato planting material production to the point where they become viable seed?
Okay. Chebet, maybe we can rephrase it a bit. How expensive is this or are these technologies in terms of producing seed potato?
Okay, yeah, I can answer that. These techniques are expensive in terms of the infrastructure. That is, they're setting up the tissue culture laboratories, the hydroponics. However, in terms of productivity, like when you check them in terms of productivity, the production is quite high as compared to a farmer who is using the conventional seed production method. And also in terms of the quantity is quite high. So if you compare in terms of cost and production, the productivity in terms of using these techniques is quite high. However, the use of conventional seed production method is cheaper than these methods.
So in effect, for a farmer to buy, let's say, 500 kilograms of seed potato from the improved technologies, it would cost him a lot more.
Yes, but the productivity will be quite high.
Okay.
And also the chances of disease spreading will be like 0%.
Much less, yeah. Okay.
Yeah.
Okay. If there are any other questions on the chat, you'll be able to look through and respond to them. Thank you for your presentation. That's quite an true innovation in our areas of crop production, particularly in potato. And we appreciate the kind of work that you are doing and the kind of opportunities and challenges that you are highlighting, even as we develop these new technologies. Now, that brings us to the end of our presentation this afternoon for the morning session. Now, for all the participants, you need to go out of this room to the general room so that we can carry out what we call a poll, or we evaluate the session. So kindly make sure that you carry out an evaluation. Just a minute, I have something else. Do we have the time? That's the issue. It's okay. Okay, we have a little bit of information. We have Duncan, who is supposed to present. Our session was over really, but because we have made good use of time, we can give Duncan 10 minutes to make his presentation. Duncan, are you in? Hello, Duncan? Okay, if Duncan is not able to present right now, there's still a slot in the afternoon session at 4:45. Okay, Duncan?
Is it Duncan or Gidraf?
No, there is Duncan, but there's also Gidraf Okeo. Who is available? Who is available right now? Gidraf or Duncan?
Yes, I'm available, Gidraf.
Okay. If you're available, you're welcome to present. You have 10 minutes presentation and about 3 minutes for responding to questions. Welcome.
Let me share.
Okay, share it.
You can see my screen?
Not yet. Okay, there it comes.
Okay, thank you for giving me the time. Though the communication was given that I would represent last year at 5, but thank you for accommodating me.
It's okay.
I'm Gidra Fonduro Okeo, PhD crop protection student, and my supervisors are Dr. Miriam Charimbo, Jane Nyanga, and Dr. Thiago Mendes. So I was carrying out a research on antibacterial activity of 4 plant species, that is guava, moringa, and campo grass, against bacterial root of potato.
The slides on slide mode.
Okay, thank you. So we find that bacterial will cause Alsonia is the second most biotic factor affecting potato production in most fields. And it is taken after red flight, which is a major challenge. And around 2014, there was some naming, and we find that now the renaming classified as the Pseudocerulosterium species, the phylotype 1 and 2, as the most species affecting the solanacea crops, mainly potato, leading to high yield losses. So we find that approximately globally, over 1.7 million acres are affected globally, resulting into high losses estimated as more than $950 million US. And the key countries affected are Kenya, China, Uganda, Bangladesh, among others. So in Kenya, over 70% of potato farms, actually solanacea farms, are infested by bacterial wilt, and this has resulted into yield losses ranging between 50 to 100% if they are left uncontrolled. And the key challenge that comes in is that bacterial wilt is a bacterial disease, and mostly we don't have registered pesticides that are geared towards management of bacterial diseases. For instance, when you go to the PCPB website, you'll find that we only have around 7 commercialized products for against control of bacterial wilt. But out of the 7, most of them are used as fumigants, which requires a lot of attention before they are used. And then that now gives us the opportunity to come in with environmental eco-friendly management options, where now we come in with botanicals. But that area has not been exploited much, because out of the 7, you'll find that only 2 are botanicals, and they have same active ingredient, that is chlorpheniramine. So this study was aimed at exploiting how can we evaluate other plant extracts that can have antibacterial activity and integrate them in the market in future as part of integrated bacterial wilt management option. So we started by isolating and then characterizing the pathogen that we were to use in the lab. Then we also collected different plant species that we were to use, and after drying them, then we followed a procedure where we weighed 10 grams and dissolved the 10 grams into 100 ml of 2 different solvents. We used acetone, an unpolar solvent, and ethanol as a polar solvent. Then we dissolved them for 28 hours with regular shaking. Then after that, we filtered and we centrifuged. And then the filtrate, we concentrated this filtrate and then air dried the paste for overnight, and we weighed and recorded the weight of the extracted weights. Then we now organized a CRD experiment in the lab, where we had 9 treatments as outlined there. Then for those that worked well, we moved further to actually assess their minimum inhibitory concentration. That is, if we diluted further, at what point was the efficacy level of the most effective, at what point was the efficacy level of the most effective plant extract reducing? So we collected data on growth inhibition zones, and then the data was subjected to analysis of variance using R version 4.10, and the means were separated using 2 key HSD at level 5, 0.05. So from the results, we find that the bacterial isolate that we isolated, when we did the identification test, we find that one, it belongs to a gram-negative class of plant pathogenic bacteria, and from the photo you can see we did potassium test, and we also looked at the microscopy test that revealed that, but we didn't stop at that point. We wanted to see at what biovar does it belong. So we also did different carbohydrate fermentation test, and that revealed that the biovar that we were using was biovar 3 race 1. So we moved again now to do the in vitro assay, and before we went to in vitro assay, we had to see the different yields of the extracts, and we found that in general, ethanol, which is a polar solvent, had high extracts as compared to acetone, extract yield as compared to acetone, but for seeds, for moringa seed, we find that acetone had high yield as compared to ethanol. Also, we can see from the pH, because we measured the pH, and we realized that the pH was towards neutral. But when we looked at the result that we'll get as we proceed, we find that the antibacterial activity actually increased with increased acidity of the plant samples. So when you look at the analyzed result, we find that pelargonium, that is Pelargonium cucullatum leaves, worked well, followed by guava, and Pelargonium even worked well than the already registered compounds in the market, that is Enrich, which is a conventional pesticide, and Kobe, which is a botanical pesticide registered in the market. Then when we move further now, we find that generally, we find that when we did the minimum in visual concentration, we find that for both Pelargonium cucullatum and Psidium indicium, we find that their minimum in visual concentration started at 6.5 milligrams per ml of DMSO. So in conclusion, we find that the two plant extracts are potential candidates against management of bacterial wilt, but now we need to do further research so that we assess whether it can work well. Both the results that we got in vitro, we can get it in vivo and under field conditions. Also, it also posed a challenge of trying to do the phytochemical analysis so that we find that what are the phytochemical constituents that have antibacterial activity against the target pathogen. Then we now, the authors would want to acknowledge CSAM for funding the research, and the Food Microbiology Lab at the University of Nairobi for allowing us to conduct the studies in their lab. Thank you.
So thank you very much, Gidraf, for that very nice presentation, and you've done it quite timely. At this time, we want to give you an opportunity to respond to one or two questions.
Yes, you can quickly respond to one question is, what do you mean by...
Chair, we have questions.
Yes, Professor Mulwa, you can go on. You can ask the question, Professor Mulwa.
Professor Mulwa?
Hello?
We can hear you, Gidraf. Professor Mulwa has raised his hand. We are waiting for him to ask the question.
Maybe, okay, can I ask you a question?
Yes.
At one point, you're talking of using DMSO as one of the solvents, huh?
Yes.
How did you delineate the effect of DMSO on the bacterium?
Now, from the literature, I found that the effect of DMSO on bacteria increases with the increased concentration. But below 1% and below, it doesn't have effect on bacteria. And if you look at the results, we use it as one of the negative controls, and the other negative control was distilled water.
Okay.
Okay.
All right.
And then,
proceed.
On the same breath,
yes,
you did extracts, and you are talking about a percent yield extract. What
yes.
is that?
Percent yield extract is, once we dissolved, then we had to concentrate. Actually, we had to evaporate the solvent so that we remain with the pastes. So those pastes are what we weighed to see that each extract, what amount of yield were we getting from each extract. That's what I mean by percent yield extract.
And that's what I was worried, because you talked of the paste.
Yes.
How did you know at this level this paste is concentrated enough? Why wouldn't you evaporate get the solid material. Then you reconstitute in distill.
What I did, I evaporated to get the solid material, which I call the paste. Then now we
And if it is solid material, please change. You say you concentrated it to
paste.
Get that solid stuff. Because if you say paste,
yes,
it has another meaning. Because you didn't measure the pasting properties of the material.
Okay. Then after that, we reconstituted 100 milligrams into 1 ml of DMSO, 1% DMSO.
Thank you. Gidraf, you can respond to
You mean you use DMSO and then put it in distilled water now?
We used DMSO as a solvent. Yes. And distilled water we also used as a negative control.
Okay.
Yes.
For the sake of
Looks
time,
like it's only the two of us. What's happening?
we can hear you, Professor.
We can hear you. We can hear you.
Can hear you. We are just listening, but for the sake of time, we allow Gidraf.
We are listening. We are listening. And we are also asking.
Where is the chair?
We are.
The chair is here, and we have 1 minute, please. Order. Order, please.
How can a farmer distinguish between bacterial wilt and fusarium wilt symptoms on the farm? So the rapid easier way that you can use is a whoosh test. But also, if you look at a fusarium wilted potato, when you uproot and cut the stem, you'll find that the one that has fusarium infection is hollow, while the one that is bacterial wilt infected, you'll find it is contact, but it is turning brown to black at the base.
Okay, thank you very much for the interest that this has generated. It's quite an interesting study with a lot of potential. Now, for the sake of time, we request that Gidraf, you look at the chat box and respond to those questions that are there. Otherwise, we want to encourage all the participants to go out of the breakout rooms into the main room and carry out the evaluation. Thank you very much for your attention and for keeping with us throughout this morning session. Thank you.